Hi, welcome to the Bioinformatics chat. After hesitating for a really long time, I finally decided to launch a Patreon for the podcast. With the war going on and me serving in the army, honestly, it has never been as difficult to produce the podcast as it is right now. And as we're heading towards the winter, it's going to become even more challenging because the Russia will probably continue its last year's tactic of striking our power plants, substations, and other energy infrastructure. So I hope that your financial support would both help me practically, but also give me a bit of extra motivation to keep producing the podcast in these circumstances. By the way, I set it up so that the payments are per episode instead of monthly. So if it happens so that I'm unable to produce episodes for a long time, you won't be charged. There are also some small perks that come with the different tiers. So please check it out. The address is patreon.com/buyinfochat, and you can find the link in the show notes or on the website. And if you do decide to join, that would really mean a lot to me. Okay, now a bit of news from Ukraine. There was no shortage of airstrikes since the last episode. One of them affected me personally. I can't really share many details about it, but I'm fine, I'm not injured or anything. What I do want to tell you about is the June airstrike on Kramatorsk, which I already talked about a couple of episodes ago. But what I didn't know at the time was that a talented Ukrainian writer and reporter, Victoria Amelina, was severely injured in that attack and died in the hospital just 4 days later. There's a very interesting podcast by The Guardian about Victoria. In particular, her search for a hidden diary of her fellow writer, Volodymyr Vakulenko, who was kidnapped and executed by the Russian soldiers. I'll put the link to that podcast in the show notes. And as I said, it's quite interesting. I encourage you to listen to it. But here I will only include the last bit of that episode, which I found particularly moving.
Victoria's death leaves behind her husband and her son. How did it feel when you found out that she'd gone?
Well, this isn't about me, and the fact is that a lot of people were terribly, terribly devastated and furious, you know? I think for myself, because I'm a cultural journalist and I've sort of stumbled into writing about Ukraine, I hadn't really taken into consideration that I would become friends with somebody and then they would die. I mean, even though that makes no sense because it's a war, people die all the time. So I felt what a huge loss she was because, you know, her literary career was just beginning to really blossom. She had so many books to write. She was such an asset to Ukraine, actually, in all the work she was doing around civil liberties, justice, war crimes. She was an excellent woman. And I felt absolutely distraught when I discovered that she died.
Please join me for a moment of silence for Victoria Amelina, Volodymyr Vakulenko, and many other talented Ukrainians murdered by Russia. Slava Ukraini. And now let's talk about bioinformatics. My guest today is David Dylus. David is a senior scientist at Roche now, but in this episode we're talking about the work he did while being a postdoc at the University of Lausanne. This work is called Read2Tree, and it's a tool for inferring phylogenetic trees from raw reads. So I asked David how he became interested in phylogenetics.
So when I was doing my PhD thesis, I was working at the University College of London with Paola Oliveri and on brittle stars and the evolution of gene regulatory networks. And there we found that there is a specific gene that was required in order to initiate the construction of a larval skeleton. And in order to prove that really we are talking about the same gene in two species that have shared a last common ancestor 500 million years ago, we needed to build a phylogenetic tree. So that was the first time that I got around this, and it really made me interested in, okay, how can we really figure out whether two genes are actually the same that we are talking about, whether we are actually talking about the relationship between species in a particular way. So that's how I kind of got exposed to phylogenetic trees, and that's why I also wanted to pursue it further.
Mm-hmm. And so when you started this postdoc at the University of Lausanne, did you start it specifically with the intention of looking into phylogenetic trees?
Yes, that's correct. So I started my postdoc to, initially we wanted to build something completely different. We wanted to build like a tree binning method where you would build individual phylogenetic trees, not for species, but for groups of genes, and then see how you can bind them together to build a species tree. But we kind of quickly got into a completely different path by being introduced by our collaborator Fritz Sedlazeck to his. He developed a method for mapping long reads to sequences, and from that moment on, I kind of started to okay, maybe we can use this method to speed up the construction of phylogenetic trees, because this was always something that was a bit cumbersome. If you want to build an extremely good phylogenetic tree, you would need to use a lot of species, so a good sampling, a lot of genes, because you need to have a lot of genes, so you need to have an extremely long multiple sequence alignment, and then comes the crazy part. You can use extremely fast methods that are unreliable. They are reliable, but not to the same degree of precision. Or you can use like Bayesian methods that are extremely good and give you an extremely nice sampling of the whole space of phylogenetic trees, but they are extremely slow, right? So I was thinking, okay, maybe these two things, it's kind of like hard to change because they are extremely well established. But what if we come up with a faster way of actually getting the input data ready for these methods, right? So that's where the whole idea then of Read2Tree came from, where we were thinking, okay, to build a tree, what we need to do is actually we need to go get samples, we need to sequence them, then we need to take the reads and filter them, then we need to build an assembly, then we need to annotate them, we need to compute some orthology in which we compare all the genes that we extracted against all the other genes from other species in order to then later build the alignment, and so on and so forth. And all these steps take an crazy amount of time, right? So we thought about if we could sidestep all the steps by using a mapping approach and then speed up the whole process, and then very quickly immediately start with the inference of the phylogenetic trees, right? So this was the whole idea of this project.
And so when you say that these steps take a lot of time, I think, and correct me if I'm wrong, but there are actually like two kinds of times that we're talking about. It's like human time for someone to sit down and write the pipeline and download, install different tools, figure out like how to run them, and then there's actual computer time to run the programs.
That's absolutely correct, right? So if you think about the multi-step process, you can either build an extremely nice pipeline, with tools like SnakeMake or Nextflow or whatever, right? And then it will work, but you need to work out, for Illumina reads, I need a different assembly method that I need for Oxford Nanopore reads or for PacBio reads, right? So you have to account for all these factors. Also, quality control works differently for all of these methods. And then later, maybe once you have an assembly, okay, annotation, maybe then you can use, Augustus or something like this to annotate and do some protein predictions. And from that moment on, again, once you have the proteins and the genes, then you can use an orthology prediction tool that will, group the genes that you inferred into buckets of genes that we already know. So that's kind of like, we would know, okay, this is that gene that belongs to this group of gene. And this computation, so all these steps take a lot of human time to figure out, which tools to use, what is currently the best tool as well, right? We have tons of benchmarking papers available to check for those. And then to set it all up and then to make it work, right? So you would have to do this for each species separately, for each technology separately. And then finally, also the computation time is massive, right? If you want to do an all versus all computation where you have 30,000 genes and you want to compare it to, let's say, 20 species where each species has 30,000 genes, the complexity explodes, right? So you would have to use a high performance cluster and make all these computations happen so that you actually get to a point where you can infer a phylogenetic tree, right? So that's and this computation time is extremely computationally intensive, right? Maybe, nowadays assemblies and so on, the steps can be done quite quickly depending on the tool and the precision that you're aiming for. But then, like, once you get to the point where you have to infer homology and orthology, then it becomes extremely computationally expensive, right? So that would be then the other aspect that you mentioned.
You kind of gave us a breakdown of the different steps that you have to go through when building a phylogenetic tree. But let's maybe spend a bit more time on that and discuss like each step. Why is it necessary? So can't you simply do like multiple sequence alignment and build a phylogenetic tree on that? Or like, can you talk about why you need those extra steps like protein prediction, like annotation? What do they actually do and what do they contribute?
So if you have a genome, right? And let's think like, you have a genome of a human and of an ape, right? Multiple sequence alignment will be extremely difficult because it's the genes are not placed in the right place, and the things that you can actually align extremely well to each other are genes, right? They are similar to each other. The rest of the genome, the non-coding regions, I think that will be extremely difficult to align, if not even impossible to align correctly, right? So you can imagine you have like stretches where the genes are placed in different locations, right? So how would you kind of like put this all together, right? You would kind of have to figure out. So that's why usually when we construct phylogenetic trees, we actually we first want to know which are the genes that are homologous across the species for which we want to construct the phylogenetic tree with, right? That's why we do this, orthology prediction, homology inference. And that would just basically take out the genes and then, okay, I know, okay, this is one gene family. And then I do an alignment of that particular gene family, right? And then I do it for all the gene families for which I find, close genes in the genome with. And then I have a lot of those and I concatenate them. And this is then my input matrix. The alignments of all these individual genes from the genome, right? So otherwise it would be extremely difficult to do, if not impossible, depending on, how distant the species are and so on and so forth.
So that's the step that you call annotation, right? And then there is the protein prediction. So do you have to sort of translate the nucleotide sequence to the amino acid sequence? What does that accomplish?
So that's correct, right? So if you have a genome, then there are usually we have in the genome we have exons, right? So we want to figure out, which are the start and ending point of exons, and if we put them all together, then we have a consistent protein, right? That has different isoforms. And that's exactly, so the protein prediction would kind of like go through the genome and check, using some like maybe pre-trained model, okay, which are the sequences that I know of. Oh, this looks like an interesting part. So it would cut it out and it would stitch it together and kind of form a protein prediction. So I'm not an expert in these methods, I have to say, right? It might be like this is quite an intuitive explanation. I bet that there are much more intricacies and more sophisticated steps in there. But basically, that's how you kind of want to get this, sequence stretches that represent a whole gene that can be translated into a consistent protein where you don't have like a stop codon in the center or something like this, right?
Okay, but like, why is it important? Is it because the axons are thought to be under more evolutionary pressure, and so there is more signal there, as opposed to like introns that, may mutate more chaotically?
That's correct. I think that's kind of a lot of then in the step at the very end, right? When we want to infer phylogenetic trees, right? We need to again use some type of evolutionary model that describes, probabilities of things switching, right? This is also something to do with like synonymous changes or non-synonymous changes. If you make a DNA model, and with protein models, then again you have probabilities of a protein, with a certain degree will to change to another, like an amino acid, to change into another amino acid in a protein, right? So you would model this type of probabilities of change, and you would use these probabilities in order to infer then later on the phylogenetic tree, right?
Okay, that makes sense. And so you were a little bit annoyed with the complexity of this whole pipeline. And so what were your thoughts? Like, what did you decide to simplify or to throw away?
So that's exactly, so here's the thing. In the beginning, it's kind of annoying, but it's also kind of fun, right, to do all these different steps, because you learn a lot about the intrinsicies of building a genome and annotating and predicting proteins and all these things. So it's kind of cool. But I was thinking, what if somebody has a lot of sequences from species and doesn't have the time to do that, or would really quickly need to understand what's happening there, like in the case of COVID, right? So in that case, we decided to do something where you would need two sources of inputs. So one is the raw sequencing read, which can come from any technology, and the other input that you would need is already a predefined orthologous groups. So these are gene families where we have, for instance, for like some alcohol dehydrogenase, which is one big gene family, would have all the genes that we find in different species that are capable of doing that, right? And we would use these gene families for some species that we pre-select as input, right? So in the example in the paper, what we are doing is we take 30 species in the tree of life, in the metazone tree of life, with different animals, ranging from human, mouse, to also sea urchins and even more distant species. So we take these, and then we have all the species in that list, but we don't have the mouse species, and we try to map that mouse species in there, and then we can use this in order to check how accurately we are mapping, right? Because we know where the mouse is placed in that tree. It's placed next to the rat, right? So that's kind of like the first experiment that we did, right? In the paper, by taking only raw sequencing read, we know the phylogeny, so we take the gene families from it, and then we just make a mapping step that puts the reads against the different genes and reconstructs your sequences from it, and we use this as an input for the tree inference, and then we check how good is our tree. Is it the same as it was before, or is it different, right? And we vary certain parameters to really make sure that we touch the edges of what's possible with a tool like this.
So you mentioned orthologous groups. Can you talk in some more detail what they are and where do they come from?
Okay, so orthologous groups, this is every time when I mention like a gene family, right, it's a more general term for an orthologous group, right? So we animals share a lot of genes that we have in common. Some genes are extremely important because these genes define some basic developmental mechanisms, for instance, right? So if you would look for this specific gene across, for instance, all the mammals, you would find that all these mammals share that particular gene, right? And this, to kind of computationally find them, we use, ortholog inference algorithms to this. And this group, of if you put all the different species together that share that exact same gene, that group would be called an orthologous group, right? So they are the genes are orthologs to each other, right? There's a bit more a fine-grained definition to this, right? And there's some nice YouTube videos that you can look up to where you can find a bit more in-depth explanation to this. But it's basically, in simple terms, it's kind of like a gene family, where you look for the same gene across many different species and you put them together, and that's what we define an orthologous group.
Okay, but computationally, inside a computer, is that like a single consensus sequence, or is it like many sequences that represent the variants of this gene? How is it represented?
So in the computer, we represent it as a set of sequences, right? So there are many sequences together, and when you build a multiple sequence alignment, you will see long stretches of things that are exactly the same, and then some mutations or some deletions or insertions in depending on the species, right? But the basic functionality of that gene will be extremely similar. And for our tool, we don't use a single consensus sequence. We actually use all the sequences for the species that we pre-select. This kind of implies that you have a little bit of a prior knowledge of what you're actually sequencing there, right? So if I go into the field and I find like a thing that looks like a bee, right? And I don't know the species, right? But I would be extremely interested in, to which is the closest bee that this represents to. I would go to the orthologous matrix browser that my boss developed. I would select, a lot of bee species from there and maybe a couple of flies or something like this to have a nice out group. And then I would use this as an input data set, right? And then I would take that sample that I collected from the bee, sequence it and get the reads. And I would use these two input sources and see where this bee is actually placed in the phylogenetic tree.
Let's take your bee example. I like that example. So you would go to the orthologous matrix. Maybe talk a little bit about what that is, that resource.
Yeah, this is a database that has extremely accurate pre-computed orthologous groups, right? So, for instance, if you are looking for a specific gene and you want to understand which other species have that particular gene, in the orthologous matrix browser, you will find that information. And you will also find information about, for instance, whether that gene, has independently duplicated in a specific species or not. Where is it present and where is it absent? Is there another gene with a similar function? All this type of information you can find in that orthologous matrix browser web resource. Yeah.
And so what would happen if the gene was duplicated in a species? You would have like two different orthologous groups with a very similar sequence or something?
So that's now it gets a little bit tricky, right? that's now the interesting part. So if you have a duplication, then you have from the same gene a very similar gene, right? And then you would have to. So there are methods where you can kind of build. That's something that we call hierarchical orthologous groups, HOGs, right? Where you would store this information, where you could then look up whether, for a gene of your interest, you have many duplications in a particular species, right? Because you can imagine, for instance, we might have a single copy of that gene, but maybe the ape has two copies of that gene. And then, there's also a bit of debate of, whether a paralogous gene can take over the gene function. So you could imagine an experiment where, you knock out the original gene and you leave that paralog there, right? The duplicated gene. Will it just take over the functionality from the gene before, or will it not? What's happening? So, but this is quite difficult. For our tool, what we try to use is kind of only the single copy orthologs, so the genes that most likely are connected to each other. So if you imagine, you would put them in a network, it's a tightly connected network where, the distances between that one gene from that one species to all the other species that have that same gene are extremely tight knit and close together, right? So that would be the input that we would use for Read2Tree.
So in your B example, you would go to the ortholog matrix website, you would download those orthologous groups, and you would sequence some DNA from your B that you're interested in, right? If you wanted to build a tree, so you would need other species as well. Would you have to go get some real samples too, or can you use maybe some reference genomes?
So that's the thing. From the orthologous matrix browser, we already get the references, right? Because we selected some Bs from there as well, right? So we get already the references, and we get them already in the stage that we need them to be in order for us later to do the tree inference, right?
So your tool would get enough information to automatically have like phylogenetic tree leaves for those species that you selected, and is only the new leaf with your sequence trees that would have to be placed in that tree.
Exactly. So this for that particular scenario, that works quite well, and we shown in the paper that if we then take, for instance, if we try to place a B within another B, that's extremely simple task because the reference data sets are very close to each other, right? So it would very quickly find nice things to match the reads on. But now think, actually we have a bee, but the closest next species would be a mouse, right? That's quite distant already, right? Would we be able to place still the bee, in the correct position in that tree? And we have shown in that paper that we can do that quite accurately up to like maybe 500 million years of distance, right? So even if the species is extremely distant, we can still use the genetic information from the orthologous groups and from the reads that we have to place a species quite accurately into that tree, right? And this is quite cool. And we compared with an assembly-based approach using similar thing, and we see that our tool is doing either a similar or slightly better job than the standard approach.
And to be clear, your tool by itself does not infer those distances, right? Or topologies. You actually call to a third-party tool to do that stuff. But the
Absolutely.
difference is in the inputs to that tool.
Exactly. That's correct. So we kind of make this step faster to get to the input.
You also had an example with the COVID virus, right? With SARS-CoV-2. And so let's say we are in 2019, 2020, when COVID just emerged, and we don't have any ortholog groups for COVID, right? We're dealing with a new virus. Are there entries in the ortholog matrix for other kind of viruses, for coronaviruses?
Yes, that's correct. So we have entries in there is actually a separate instance, which is a virus-specific instance of this orthology matrix browser. And in that paper, we also kind of reconstructed the phylogenetic tree using references that were, I don't think, COVID. So there were other viruses, respiratory viruses that we used. And there, what we did is, you take a backbone of reference viruses, and you take many different sequence reads from many strains that you sampled from humans, right? And you map them, all of them. You construct a multiple sequence alignment, and now when we build the tree, we actually see that we can accurately discover in this tree the right variants that we also saw, right? So, we had an alpha variant, an omega variant, a delta variant, all these things we can see, right, if we do that.
Mm-hmm.
So you don't necessarily need to have a COVID virus as a reference in your backbone, and you will still, do a good job of getting the right tree.
That's amazing. And so the saved human time, like work time, is obvious because you don't have to go through, writing a pipeline. Your tool is essentially a pipeline, right? You just give it inputs and it calls the right tools and it gives you the output. But what is, in your experience, the saved computer time? Like, how much faster is your tool than the sort of standard pipelines?
So we tested a lot of different parameters, right? Like, how many reference species you have, what's the read coverage that you have, and so on and so forth. And in the paper, I did a computational assessment of the time savings that we can have, and it's 10 to 100-fold faster to get the tree, using our method, where you can imagine, all these steps before we start the phylogeny inference are the ones that are computationally intensive, right? So I compare those steps, and it turns out that, depending on the the read origin, whether it's transcriptomic or genomic, the technology, the coverage level, the distance to the closest neighbor in the backbone, all these parameters are assessed, and using all these parameters, we see that we consistently are 10 to 100-fold faster than what is out there.
And in absolute numbers, how long does it, like, is it minutes or hours or days?
So, okay, that's again a tricky question, right? Let's say you have a 10x a B, right? And you have maybe, let's say, 15 species that you want to map it to. It might take an hour or something like this to run on a regular computer, right? Because what it would do, it would go, from it would map the reads to all the genes that you have in this 15 reference species, and then give you the output matrix, and the tree will direct the Read2Tree tool will directly also use IQ tree at the end to give you a first phylogenetic tree, right? But you will have the multiple sequence alignment as well to do your own assessment.
Mm-hmm. Right. And how would you say that time is split between the preparatory phases and the actual tree inference?
So the pipeline, IQ3 is extremely fast. So the pipeline takes longer than what IQ3 does. Yes. So my pipeline would take, if it's like a short, 15 species and we map one species on it, it would take maybe like half an hour on my tool. Again, that depends on a lot of different parameters, right? And then IQ3 on an extremely fast setting would take maybe 5 minutes to give you a tree without bootstrapping. So without giving you a confidence whether for a specific split in the tree, right?
So IQ3, what paradigm does it belong to? So from what I remember, there are like Bayesian tools and maximum likelihood tools.
Maximum
and some
likelihood.
Other... Okay.
It's a maximum likelihood, and I think it's a really, really, really cool phylogenetic inference tool because it's fast and the trees actually look quite accurate, right? So it does an amazing job, and it's very well maintained and developed. It's a really cool tool.
Okay, awesome. So now that we've discussed this from a user perspective, let's now move to your perspective, the developer perspective, right? How does Read2Tree actually work under the hood?
Okay, so what we basically do is we use the fact that we have the orthologous groups and the capabilities of the OMA browser to reobtain the exact DNA sequences for the protein sequences that we downloaded. And with the DNA sequences, this allows us then to, take the reads that we have and map them against the DNA sequences. Like you have kind of like an artificial genome, if you like.
Why do you start with protein sequences?
So, okay, this is an intrinsicy of the OMA browser, right? If you download orthologous groups, you get the things that you get downloaded is protein sequences, because that's what we use as an input to build phylogenetic trees. We don't use the DNA sequence, we use the protein sequence.
So like the data you obtain from the orthology matrix resource are protein sequences.
Exactly.
And because you have raw reads, which are nucleotide reads, yeah, you have to get them to a common denominator.
Exactly. So that's why, and the cool thing is, so you have the protein sequences, but you also have the corresponding DNA sequence. So if I build an alignment, right, of the protein sequence, which is an easier task to do because they're on the protein level much higher conserved than on a DNA level. then later on, when I map to the DNA, it's much easier for me to place the sequence in that alignment because I pre-build it using the protein sequences, right? So it makes this task much faster. So I don't have to recompute an alignment. I just have to place it back, right, in what I already have, because I have the DNA and the protein sequences. And for the mapping step, so you take the reads, the nucleotide reads, and you use this tool that Fritz Sedlazeck developed, which is called NextGenMap, and that maps the reads against the DNA sequences. And now you have two things that you have to solve. One is I have the DNA sequences for one gene, but for multiple species. So which one do I choose as being the best representative for the species that I try to place into the phylogenetic tree? This is one issue. So you have to, there I defined some type of metric in order to decide which would be the best representative.
So is that done for each read separately?
No, so this is done after all the reads are mapped, right? So you have all the reads mapped. So I have a file that tells me for each nucleotide position, given the reference, I know the nucleotides where they are, and then I take from there. I have to decide, okay, is that a good representation? Because when I map the reads against the reference at the same location, I can have many. If the location is, for instance, an A, I can have many A's, but maybe one is a T, or maybe I have a 50-50 thing or a 50-40. So I have to use some 60-40, or I have to use some type of metric to decide, okay, at this position I have a ACGT, right? Which nucleotide to choose from.
Yeah, so you have actually many references, right? It's not just, so you map all the reads against all the references.
Exactly. And then it's a matter of choice, right? So I think the reason why Read2Tree performs so well is because the metrics, how we choose then later on the right representative sequence for our species, they work quite well, right? And they are extremely simple. It's just a matter of what is the maximum encountered nucleotide for the position, and then later on, what is the longest extracted sequence that we find, right? Because we have the many references. And that's how we decide which sequence to choose for our species. And by doing that, we get quite good results.
So do you try to, you don't try to pick a single winning reference, but instead you construct a completely new sequence?
Exactly.
Is that correct? From your reads?
Yes.
So you do sort of a per gene assembly or per gene mapping?
That's maybe it's something like a reference guided assembly, right? Where you have a reference and you have the nucleotides, and I use both information to extract for that gene a new gene sequence for the species that I'm trying to place. So it's it's not like a complete assembly. So it doesn't do, for instance, what you could think of is you bin all the sequences, and then you use an assembly tool to take the bin sequences to reconstruct you a new sequence, right? By doing a proper assembly, whether it's a graph assembly or any other assembly method. I tried this as well, but this didn't work so well as just taking the mapping and then using the mapping information to extract the right sequence for that particular gene. It's kind of surprising, the methodology is extremely simple, but simplicity allows for speed, right? So as if you have a simple way of getting stuff done and it doesn't compromise accuracy, then we can increase the computational efficiency with it.
So do you have to pick a single reference that you use as a guide to construct your new consensus sequence, or do you look at all the references at the same time? Because the position may differ, right?
Yeah, that's correct. So for all the references that I have a set of orthologous groups. Okay, so these are gene families. Let's take, for example, a single gene family. For that gene family, I have from every species a sequence, a DNA sequence. Yeah. So this is my gene for every species of the backbone.
The length can differ, right?
Exactly. The length can differ. Absolutely correct. That's absolutely correct. And now what I do, I take the reads that I have and I map them against every species in that orthologous group. So every gene sequence in that orthologous group. So I will have, if I have 15 of those, 15 potential candidates to choose from to decide, for the single species that I want to have. So then I check, what is the most likely sequence that represents my species that I try to place by what is the best coverage, and by secondly, what is the longest reconstructed sequence? So how many base pairs do I find where I can have a nucleotide placed during the mapping step, right? And these two criteria then decide, okay, actually, it turns out that for that gene family, when I try to place the bee, the Drosophila, the fly sequence, was much better representative for that gene family than actually any of the bee sequences. And then I would use, what I reconstruct based, so the local assembly, reference guide assembly on top of that Drosophila sequence.
Right. So that's what I meant. So you do pick like a single winning
Yes.
Gene and use that as a guide.
Yes, indeed.
But that winning sequence can be different for different genes, right? For one gene, it could be Drosophila, and for another, it could be, I don't know, a tick.
Yes, absolutely. That's correct. That's how, right? Because you would expect, so if I look at statistics, right, from the tests that I've done, in many cases it will pick, like for instance, if we stay with the bee example, it will pick another bee species, right, that we have. But there are cases in which it will actually pick a more distant ancestor. And if I look and compare this, it's actually representing it. This sequence that I reconstruct is better representing what you would get out from just, doing an assembly, right? We do that in the paper as well. So I take the reconstructed genes that we do from this, reference guided assembly approach that we do there, right, from this mapping approach, and I cross-check them with, what the original assembly derived sequence is to check for similarity, right? And you see that actually with this approach, we are capable of extracting gene sequences that are extremely similar to if you would do a regular assembly approach, right?
Are you worried though that can introduce bias? Like if you base your guided assembly on, let's say, Drosophila, then in the phylogenetics tree, the inferred distance to Drosophila would be shorter than otherwise warranted.
So that's true, but we are using, so in this paper, we recommend to use many orthologous groups, right? So if you go across many different orthologous groups, there might be, 5% where the Drosophila was picked. But in the other 95%, the bee was picked, another species of bee, right? So over many orthologous groups, the bias kind of diminishes, right? And we capture nicely the variation that we expect to see in a species like that.
No, but exactly. Like, I mean, okay, let's say you have another bee species there, and in 95% of the time, that bee species reference would be picked. And so the similarity of your new species to the bee species would get pushed, towards that bee species. So it would appear more similar than it actually is, just because that bee species was picked as the closest, maybe fairly picked as the closest reference, but still that short distance would appear even shorter because that's used as a reference.
That's correct. So I would say, so this comes into an interesting part, right? So from a point of placement, right, where we are interested, is it the correct neighbor? We are safe. But if we are interested in how closely we are related to that closest neighbor, then it gets a bit more tricky, right? Then this, you're absolutely correct. We might, the branch length between the two, that might be not as accurate as if you would do it, with a standard approach, which is less biased, right? Because you are absolutely correct. In this case, the backbone is providing the bias to what we will see in the phylogenetic tree.
So that's it, right? Did we discuss the whole algorithm, or is there anything
No,
else left?
that's it. Once it's picked, we just put it back into the multiple sequence alignment, and then we concatenate the different orthologous group multiple sequence alignments to one big one, and that's the input for the IQ3 tool in the end.
So then does the whole tool, the whole pipeline deal only with nucleotide sequences?
So the whole pipeline deals with nucleotide sequences, but at the end, once I get to a point where I have the nucleotide sequence, I place it back into the protein alignment, right? So I'm making use of the knowledge that I know, if the closest reference that was picked is a B, I take, the conversion from that triplet of nucleotides into a protein from that particular B.
Is that because IQ3 expects a protein sequence?
So IQ3 can work with DNA or protein alignment, but I think in generally in phylogenetics, if you try to resolve phylogenetic trees over a long distance, right, you would rather use protein alignment because it's easier for us to find similarity across proteins than it is across DNA.
Can you talk a bit about the orthology matrix? Like, who maintains it? How is it built? And like, what if you wanted to extend it with your own species? How can you do that?
Okay. So the orthologous matrix, so the short is OMA, and it's also the OMA browser. This is maintained by Christophe Dessimoz group at the University of Lausanne.
Is that the same group where you were?
Exactly, it's the same group that I was in, and the individual that actually is maintaining it is Adrian Altenhoff. He's also he's the second author on that paper that we're discussing. And what's happening? So the OMA browser and this whole orthologous matrix is a highly accurate tool where you compute between each gene a Smith-Waterman alignment. So you get the global optimum alignment between two sequences, right, to construct a network, and that's kind of like how they estimate what things to put into an orthologous group by doing an all versus all. So all genes of one species against all genes of all 2,000 species that are there. Right. So this is an extremely computationally expensive undertaking, right? But on the other hand, you have extremely reliable orthologous groups.
And what if you want to extend it for your own purposes? So maybe you're working purely on coronaviruses and you want to build your own matrix just based on the different coronavirus references.
So that's possible. The algorithm is freely available, right? You can download it and you can just run it yourself. It's also extremely easy to set up. You just need to have the genes or the proteins from your sequences of interest. You put them into a folder and you just run the algorithm and it just computes everything and gives you the orthologous groups at the end.
All right, pretty cool.
Yeah, it's a very cool tool.
Is there anything else that I didn't ask you about that you wanted to talk about?
I'm quite happy that it took this whole work took an incredible amount of time to set up, to make all these experiments run, to make sure, how comparable are we and what are the edges, right? Because I'm always thinking, like, I like to use tools where I understand, when will it break? What are like the limitations of it, right? So we try to characterize this as good as we could, right, in the paper, to make sure that when people start using it, they will know, okay, it works well for these conditions. It probably, a different approach might work better for a different set of conditions, right? So you have this understanding. That was for me extremely important. And the second thing was that it should be easy to run, right? So it's a Python tool. If you have a command line, it's really easy to set up and to just make it work, right? So this for me are quite important things if you develop a tool, right?
Well, David, very cool tool. Thank you for coming on the podcast and good luck on your new job.
Thanks. Thank you very much. And thank you for doing this. It's really cool. I find this really awesome. Thanks a lot.